Today, we're extremely pleased to have Dr. Ian White presenting today on modern processing techniques in perinatal biologics. Perinatal biologics are one of the most poorly understood, but seemingly some of the most popular technologies coming onto the market, and it seems like almost every day there's a new company coming up with some derivative of a birth product. Dr. White is considered a leading expert in the field of regenerative medicine. He has over 20 years experience working with stem cells. He's published groundbreaking research in the field of regenerative medicine alongside other scientific greats like Dr. Joshua Hare, including a book chapter on the use of mesenchymal stem cells in cardiology. Dr. White's work in regenerative medicine of the heart was featured on the cover of Circulation magazine and one of the top peer-reviewed journals. I could probably go on and on. However, we're very pleased to have Dr. Ian White to discuss perinatal biologics, some of the basic science around regenerative medicine. We're going to discuss a little bit about the regulatory structure of 361 versus 351 and some of the protocols that have been working in clinics around the country, including. So, Dr. White, thank you for being with us here today. We're very excited to hear your presentation.
My pleasure, Joe. Thanks for inviting me to talk today a little bit about regenerative products that are currently available on the market and being used clinically. Background on me. I came from England about 20 years ago to work in the field of regenerative medicine and stem cell biology. I spent a couple of years at Harvard University working under Dr. Laurie Glimcher, where we studied hematopoiesis and cells of the immune system. I then went on to work at Cornell University for my PhD, working with Dr. Shaheen Rafi, who is a Howard Hughes investigator at the Ancestry Stem Cell Institute there. And my work was focused on developing techniques for ex vivo culture of hematopoietic stem cells so they could be used in regenerative medicine. And that technology has since gone on to be commercialized by Cornell University, and it's doing very well. I also worked at the University of Georgia under the mentorship of Dr. Stephen Dalton, whose pioneering work on c-Myc led to its inclusion as one of the 4 factors used in iPS cells in order to generate the stem cells from fibroblasts that Dr. Yamanaka won the Nobel Prize for. I went on to the University of Miami to work with Dr. Joshua Hare, where my interest in regeneration focused on cardiac regeneration. And from there, I founded Biopharma, which is a stem cell research and manufacturing company, and became the chief scientific officer for IMAC Regeneration Centers. And so I'm going to be talking today about some of the products that I've been working with and some of the products that are being used in the IMAC Regeneration Centers. So this video that's showing right now is really the reason why I got into regenerative medicine. What this shows is a salamander limb that's been resected, and over the course of several days, the scientist has taken photographs of the regrowth of that limb and stitched it together to form this video. And I thought this was really quite amazing how the limb of this salamander, and in fact appendages of many different species, retain this ability to regenerate even after birth, when the human body really is quite limited by our regenerative ability. That said, we do retain quite a substantial ability to regenerate. And in fact, for the first few years of our life, we actually are very robust in our ability to repair injuries and to replace lost cells. But unfortunately, what happens over time is that as our demand keeps going up for stem cells, our pool for stem cells decreases. Interestingly, this is a evolutionarily conserved feature of many different species, and in fact, you could argue all species alive on Earth require a mechanism by which to remove individuals from a population and replace them with new individuals. So in order to get new life, you have to have destruction of the old. And that process of removing individuals from a population and giving a new generation a chance is what we refer to as aging. The mechanism by which we are removed from a population is aging. And if we think about it, that's actually what we are fighting against as regenerative scientists and regenerative physicians. Everything that we see in the clinics and everything that we research and study pertains to aging. Again, aging is our progressive loss of stem cells as we progressively need more of them. And so I thought that was particularly interesting because every single tissue in our body contains either a stem cell or what I prefer to refer to as a regenerative cell. So a stem cell really is a very specific term and is typically overused. It's a bit of a misnomer. And in fact, if we use the heart as an example, recent research has demonstrated there are no stem cells, no canonical stem cells, something that we would really consider a stem cell in the heart. However, what I demonstrated in my research is that there is an ability of the heart to regenerate. Within the first few days of life, the heart can actually repair direct injuries. But those injuries and that repair is completely dependent on nerves and the signaling from nerves that communicates via the vascular endothelium to the tissues. And in fact, the regenerative tissue in the heart is actually the epicardium, which is a single layer of cells that surrounds the heart and actually mediates the regeneration. I'm going to talk a little bit more about that later when I get into my research. But when we're talking about stem cells and stem cell therapy, what we're really talking about is cellular therapy, because the word stem cell is, like I said, a bit of a misnomer. For the most part, the cells in our body that contribute to regeneration don't have the canonical features of stem cells. So here's what I was referring to earlier. As we age, our demand for stem cells goes up and the supply goes down. So what we end up with is this extremely high demand, a very low supply, and that's how we recognize the features of aging. And often that is demonstrated by multiple different types of injury that are sustained that are either acute or chronic. These are degenerative injuries as well that occur over time. A lot of the issues that we see at IMAC are things like torn ligaments and tendons, muscle injury, and these all stem from an inability of stem cells or regenerative cells to get to the areas where they need to repair tissue. And we like to refer to a lot of these tissues as white tissues. Most of the tissues that we see in the clinics are tendons and ligaments that have eroded or become damaged. And the reason they're white is because they have a very poor blood supply. And so if the pool of stem cells is already low, the number of stem cells or regenerative cells getting to that tissue, that white tissue, is going to be even lower as we age. And so what we need to do is intervene. And so we're able to use guidance and get into those tissues and supply regenerative medicine, regenerative cells. And then at the IMAC clinic, they also incorporate rehabilitation as well. So rather than just injecting stem cells or regenerative cells into a tissue, the body is actually rehabilitated too in order to make the most of that regenerative therapy. Now, we like to focus mostly on perinatal cells, perinatal products. Biopharma is a perinatal manufacturing and research facility. And the reason for that is over my 20 years, I've worked almost exclusively with perinatal products, some adult tissues as well. But it's really the perinatal tissue that retains the most potency. As you can see here from this slide, if we're looking at MSCs, a newborn has a lot more MSCs, which are a lot more potent than everybody else in the population. So by the time you're a teen, that potency has dropped off precipitously. And if we're looking at 60-year-old patients, there's really very little stem cell or regenerative cell activity. So utilizing the power of newborn tissues is sort of how we've experienced a lot of success. And this is not new. There are many groups out there that understand the importance of aging when it comes to tissues. Here's just an example of how aging in MSCs decreases with age. And you can see here that the correct nomenclature is being used. Mesenchymal stromal cells, or also mesenchymal signaling cells, is also appropriate. We're now moving away from the term mesenchymal stem cells because we in the field have agreed that the term stem cell for MSCs is not appropriate. They don't have the canonical features of a stem cell, and so we're actually now moving away from that term. So when talking about stem cells and stem cell therapy, we're not really talking about stem cells. We're talking about cellular therapy and other regenerative tissues. So just be aware of that. So as I said, there's a lot of interest right now in the power of young blood, perinatal products, and a lot of this research was born out of Harvard, where they were looking at a process called parabiosis, which is where an old mouse and a young mouse have their circulations combined, and they're able to share the same blood supply. And what they noticed was that the older mouse demonstrated phenotypes that were affiliated with a younger age, and the younger mouse started exhibiting features that were associated with being older. So it was a very interesting observation that clearly there was something in the blood that helped the old mouse to become or appear younger. And indeed, what was found that these features, these factors, whatever it was that was present, were also present in the plasma. You didn't even need the cells. And so what we're sort of discovering from there is that there are exosomes, which are the signaling particles that cells use in order to communicate. Those are present in the plasma. And in a young mouse, those exosomes contain a lot of pro-regenerative, pro-growth, anti-aging growth factors. Again, there's a lot of interest. In 2015, Johnson & Johnson spent $50 million trying to understand the magic behind parabiosis. But other groups haven't even been waiting for the research to come in. You may have heard of several companies that are buying young blood. They buy blood from teenagers, healthy sports stars that are at college. They pay them a few $1,000s for their blood, and then they give that blood to typically aging CEOs and those that can afford it. The idea of this is to take advantage of this parabiosis effect. And this is becoming surprisingly quite popular, where older people are buying young blood and having it infused into them. Not necessarily something I would recommend, because, of course, there are a lot of issues with transplantation of blood. You've got to do a lot of tests, and there are also a lot of things we don't really test for that could cause problems. So that's not necessarily the way to go. But what I want to talk about now are more legitimate sources where there is a lot of research and there's a lot of safety data, and that's the use of perinatal products for therapeutic use. Many of you are probably familiar with bone marrow and adipose as a source of cellular therapies, but we typically like to focus on the perinatal products for the reasons I mentioned earlier about their potency and their ability to really give the body a kickstart with regeneration. So here's a list of some perinatal products that I've been working with over the years. We have umbilical cord mononuclear cells, and often you might hear in the market these are referred to as umbilical cord stem cells. Again, they're not really stem cells. There are stem cells in there, but these are hematopoietic stem cells. And whenever you hear discussion about the difference between umbilical cord or perinatal products and bone marrow, and that there are no live cells in perinatal products, umbilical cord blood, but there are in bone marrow, that's pretty much nonsense. And in fact, bone marrow is essentially the same product as umbilical cord blood as far as the stem cell count and the types of cells that are present there, except that perinatal blood, the blood from umbilical cord, is extremely young, is extremely potent, and you don't get any reactivity, which is very interesting, because if you think about it, the baby and the mother have to share a blood supply, and so during the developmental period, the immune system is modulated quite heavily so that the fetus isn't rejected, and we're able to take advantage of that in our therapeutic products. So when we take these cells, they're actually immune evasive and are able to exert regenerative potential in the body. So Wharton's jelly. Well, Wharton's jelly is a product again from cord, but this is from the tissue of the cord, not from the blood. There are a lot of MSCs that are housed within the Wharton's jelly. This gelatinous like structure that holds the vein and the artery. We also have other tissues from the placenta. There are also umbilical cord PRP products, amniotic fluid, and a lot of folks in the marketplace might refer to that as amniotic fluid exosomes. That's different from other exosomes you might see in the market. Amniotic fluid exosomes are strictly perinatal exosomes from umbilical fluid that come from a heterogeneous population of cells during a period of intense growth and regeneration and immune modulation. So they're extremely potent when it comes to therapy. Other exosomes you might hear about are from adult MSCs, perhaps, that have been cultured, and those exosomes are the result of long-term culture of these MSCs. They're harvested from the media, ultracentrifuged, and then provided as a therapeutic. And these two have been demonstrated to have great effects in the clinic. So how do they work? Well, for a long time, we thought that the cells, stem cells, regenerative cells, we thought that they contributed directly to tissue regeneration by differentiating into that tissue type. But what's becoming clear now is that the cells really aren't around in the body long enough to differentiate into those different types of tissue. And so what they really do is instruct the area, the injury, the inflammation, whatever the indication is that we're trying to treat. Those stem cells or regenerative cells are able to home to the site of inflammation, and then they direct regeneration through a paracrine signaling. And paracrine signaling is essentially the production of exosomes. So those therapeutic cells go to the site of regeneration. They induce regeneration at that site by communicating with host cells, innate cells, through exosomes and instruct them how to regenerate. They induce proliferation, and in many cases, they're able to actually donate mitochondria, which is like a battery pack, and gets them going again. So you may have heard of exosomes. It's an extremely hot topic right now in perinatal research. And basically what exosomes are little tiny buds that bud off of cells and are packed with information like protein, mRNA, and miRNAs. And these are what instruct neighboring cells how to behave. The therapeutic cells are able to sense the environment, find out what's wrong, send out instructions, and the host cells are able to absorb these instructions and respond. Here's just a list of some of the things that you might find in exosomes. The list is really more comprehensive. There are many growth factors and cytokines that are present in exosomes. One of the main things that exosomes do are able to activate T regulatory cells in order to help modulate an immune response and modulate a M1 to M2 macrophage response. And there's really a lot going on with exosomes, and we're really just sort of scratching the surface of their potential at the moment. So just a quick idea of how some perinatal products are made. This is an example of how I make amniotic fluid-derived exosomes. I receive amniotic fluid from an FDA-registered collection company. I then process that product in the lab using various techniques, including centrifugation and filtration. I finish the product off with a sterilization filtration, and then I end up with a product with typically around 2 to 3 billion exosomes per cc. And again, these are a heterogeneous population, unlike those that are culture expanded from MSCs, which are a homogeneous population, the same exosomes produced all the time. This is a heterogeneous population from multiple different cell types, and I believe for that reason they are much more potent. So here's some data, and as a demonstration of why you need to do your due diligence when it comes to purchasing these products if you're going to use them therapeutically. Not all lipid vesicles are exosomes. So cells produce a lot of different vesicles that are lipid-based. You have a phospholipid bilayer that buds off off cells with all kinds of junk and goodies too. And so the important thing is to know that you're getting exosomes rather than apoptotic bodies or anything like that. So one of the last filtration steps we do is a sterile filter, which not only makes sure that the product is sterile, but also it isolates particles that are between 50 and 200 nanometers, and that's typically the size of exosomes. Other vesicles are typically much larger, and I think I have another slide here. Here's an example of where other companies may stop. On the right-hand side here, you have particles up to 400 nanometers, and now that's not going to be exosomes. That's going to be a lot of junk that you're infusing into a patient, and we have no idea how that's going to behave. So what we want is a product that actually has vesicles that are between 50 and 200 nanometers, and in fact we can do surface staining of those vesicles and demonstrate that they express markers of exosomes, and that's also something we're in the process of doing. So some clinical research and evidence. Well, here's an article that was published a few years ago now saying that humans might develop salamander-like regenerative abilities soon. So this is harking back to my initial slide with that video where the salamander was regrowing its limb, and I questioned, well, why can't humans regenerate tissue like the salamander? Well, my research sort of took off from that point, and I started to try to understand why the salamander retained this ability to regenerate and humans didn't. And one of the things I found with some established research was that a critical component of regeneration is nerves. And so what this group demonstrated here on the right is that when you resect the limb and it grows back, there's this bud on the end called a blastema. And that blastema is full of stem cells. But in order for it to actually regenerate the limb, it requires signaling from a nerve. And if you denervate that nerve, if you kill the nerve upstream, that limb actually doesn't grow back. And it's very interesting. If you look during ontogeny, during development, nerves and blood vessels have a very tight relationship. And in fact, as nerves are growing during ontogeny, if you were to change the trajectory of those nerves, blood vessels actually turn and follow them. So they're very closely associated, and blood and nerves are absolutely required in order for blood vessels to form. So of course, if we're trying to grow new tissue, one of the biggest hurdles is to grow new blood vessels into that tissue, is to reperfuse that tissue. And what we demonstrated was that if you have nerve growth, you're able to get new blood vessel growth and then tissue regeneration. Here's just an example of how some topical use of umbilical cord blood has had dramatic effects on healing in the diabetic wounds of mice. And it turns out that diabetic wounds in mice don't heal very well because they're typically denervated. Many diabetic patients have peripheral neuropathies where the nerves die back. When the nerves die, you get an issue with the blood vessels. You get a fenestration of the blood vessels where they become leaky and damaged, and then they don't perfuse the tissue correctly or appropriately or sufficiently, and so the tissue starts to die. So you get necrosis. So what's interesting here with the stem cells, applying those, you're able to get tissue regeneration through nerve growth and revascularization. So I took this a little further with my own research. This was featured in the cover of Circulation Research in 2015, and I was awarded the Best Manuscript Award for this work. And in a nutshell, what I basically showed was that the one-day-old mouse is able to regenerate the heart if you resect a portion. If you cut a small portion of the heart and replace the heart in the chest cavity and recover the mouse is able to regenerate that tissue. And what I showed was that this is mediated through the growth of nerves. If you look at B at the top there, you can see that during the regenerative process, the first thing to grow into that area are nerves. And those nerves are bringing in with them blood vessels. And those blood vessels then reperfuse the tissue, and we're able to get tissue regeneration. And what I showed in this paper was that if you denervate the heart, if you kill these sympathetic nerves that innervate the regenerating tissue, you lose the ability to regenerate the tissue. And that's what I'm showing there in C. The control, you can see after regeneration, there's a little tiny bit of scar tissue, but the tissue is regenerated. And interesting, there's a lot of lipid there. There's a lot of fat just to the right side. a lot of people used to think that fat associated with the heart was prognostic for heart disease, but it turns out that actually, at least in my opinion, fat in the heart and other injury sites is actually used by the regenerative tissue as a source of energy. Fat is very biologically active, but it's also a huge source of energy. And if you're building new cells, you need new lipid, and fat is a source of that. So that was a very interesting observation. But if you look to the right-hand side of that, what you see is that over the same time period, if you've denervated the heart, that tissue does not regenerate at all. So I was able to take that principle that there's something in the neonatal heart that allows regeneration to grow, regeneration to occur, that's not present in the adult heart. And so we started focusing down on the nerves and what's produced by nerves. And we found a couple of key factors that were very important in regeneration in vivo. And so we took those factors out. These are the sort of factors you might find in exosomes, for example, and we put them in a culture condition where a neonatal heart, a one-day-old heart, was placed in culture. And at the top there, we use canonical media, which means that we use media that's typically used to grow a monolayer of cardiomyocytes. But because this is a 3D structure, the tissue died very, very quickly. And everything is, excuse me, everything is necrosed right there. There's a very fast coagulative necrosis that occurs within 3 days. But if you look on the bottom there, when we add the factors that we discovered, we're able to keep the heart viable for extended periods of time. And in fact, you can see that the sarcomeres are intact, the nuclei are intact. We don't have here some of the other images, but we have electron microscopy showing mitochondria and other structures that are intact. What you're seeing here is the same heart in culture after 14 days, and you can see these punctate areas of beating. So typically when we take the heart out and we try to culture it, everything dies within 3 days. But in the case of this culture system, using factors that we liberated from the communication between nerves and blood vessels and the cardiac tissue, we're able to get ongoing viability of the heart and also contractility, not only demonstrating that the heart's alive, but it's also functional. So that was a very exciting observation. Here's something else very interesting. So in the industry, a lot of folks say that cellular therapy or stem cells do not integrate into tissue and do not help regeneration. It's all placebo. But what I'm demonstrating here is that I took embryonic stem cells and differentiated them into epicardium. And if you remember from before, I mentioned that the heart does not have stem cells, but its regenerative tissue is orchestrated. The regenerative process is orchestrated by epicardium, the epicardial cells that surround the heart. And so I took those epicardial cells and I dropped them into a petri dish with a heart that had several injuries. And I colored them green with DIO. It's like a GFP, so you can see the cells. And what you can see here is that exclusively the cells associated with the injury sites. And that's very, very exciting. It shows that not only do the cells remain viable, they also integrate into the sites of injury and they help mediate repair. And if we took this out longer, what we'd find is all those GFP cells disappear and they're replaced by host tissue. And so what this means and what we believe is that these therapeutic cells are instructing the local tissue how to regenerate, and then they are eventually removed from the system and replaced by host tissue. Now that's in vitro. Here's just a quick example of a cross-section, some histology showing those epicardial cells entering the injury site. But what we're really interested in is what happens in vivo, what happens inside the body. And so we go back to this neonatal mouse model where we're able to incapacitate the neonatal mouse and make an incision around the fourth intercostal. Using a modified needle, I'm able to scoop out the heart by the apex of the left ventricle. I can then resect that tiny little portion of the left ventricle, replace the heart, suture the mouse, and recover the mouse, and then watch the recovery. And what I did in this experiment was I took the same stem cells, I differentiated them into epicardium again, colored them green, and simply dripped them into the chest cavity. And what you're seeing here is after 8 days following injury, those green cells are exclusively associated with the injury site. Those green cells that I simply dripped into the chest cavity, I didn't inject them peri-infarct or anything like that. I simply dropped them into the injury site. In fact, this isn't an infarct, this is a mechanical injury. But the cells were able to find the injury themselves. This is a major feature of regenerative cells and of stem cells. They're able to find the sites of injury and incorporate and mediate repair. And that's what you're seeing here. So, okay, we've seen in vitro, we've seen in vivo experiments with mice. What about with humans? So this is an example of a patient that came through IMAC regeneration centers. She had been suffering with steroid withdrawal syndrome for 5 years. She had a skin issue that the physicians were treating with topical steroids. This led to major issues with her body as a whole. She developed this extreme reaction once she had to come off the steroids, and this covered large portions of her body, and it literally looked like she'd been dipped in a vat of hot oil. She was very much incapacitated. She was bedridden for a large part of this, and nothing in conventional medicine over those 5 years could help her. So she came by the IMAC regeneration centers, and we gave her some perinatal products. We gave her umbilical cord cells, and we also gave her exosomes, and this is 1 week later. So as you can see, she's not completely healed, but there's a dramatic change from perpetually having raw skin, exposed skin, to now certainly being on the way to healing. Again, this is her arm, and then 1 week later, you can see that it's dramatic improvements. And in fact, she's now had 4 treatments, and she's, for the first time, been able to swim in a swimming pool with her 5-year-old daughter, because her daughter had never known her to be well. She was featured in a magazine and in several news articles because of the dramatic effects that she experienced. Here's another example. I'm not sure if these videos will play, but I'm going to give it a try. So you can hear there the audio. So this gentleman was 4 years post-stroke. He was akinetic on the left side, unable to move his left side for 4 years. He received umbilical cord cells and exosomes. And then 20 minutes after receiving that, he told his wife that he had the urge to move his toes. And what you're witnessing there was him moving his toes for the first time in 4 years. Here's another video. This is him again 24 hours later. Again, previous to this treatment, he was akinetic in that left side, spastic muscle, unable to move. And again, this is 3 days later. He's able to cross his legs for the first time without the aid of his hands. And so you might be asking, well, how can this act so quickly? 20 minutes after infusion, this can't be regenerative medicine. And in fact, what you're witnessing here is the power of what we take advantage of in IMAC regeneration centers. We look to the body to heal itself, and then we contribute where we're able to contribute. So we gave this gentleman exosomes. Those exosomes were able to reduce inflammation within a 20-minute period, so much so that he was able to start sending signals from his brain to his foot and back again. Within 24 hours, he was getting so much better that he actually could then operate and flex his ankle. Again, a reduction in inflammation, probably no regeneration at this stage. But what we're doing now, several weeks after this, is rehabilitating his body. So now we've reduced inflammation, and the regenerative cells are starting to take action and repair and regenerate some of this injured tissue that was a result of the stroke. We're now rehabilitating him and teaching his body how to walk again, teaching his brain, teaching his muscles how to walk again. And now this guy is actually ambulating very well. So that's a very exciting success story. So in summary, what have we talked about here? Well, we talked about how, as regenerative scientists and regenerative physicians, really we're all in the business of anti-aging. aging is really the mechanism by which we have evolved to remove individuals from our environment and replace them with new individuals that perhaps have different genetics and can adapt to the environment different, or even move to new environments. And so as we age, we lose stem cells. This is something that can actually be measured by measuring telomeres. So telomere length is an indication of essentially your potential when it comes to stem cells. Dr. DeFreitas is an expert in this, and I know that she's a regular presenter at Boston BioLife. If you don't know, telomeres are essentially the cap ends to chromosomes. When a cell divides, the chromosomes need to be duplicated, and that duplication process requires machinery that has to sit on the chromosome and move along the chromosome, and it needs a docking port, and that docking port is the telomere. But every time it docks and reproduces the genetic material, the initial docking site is lost. And so every time it docks on a telomere, the telomere gets shorter and shorter and shorter, and to the point where it gets into the genetic material, at which point the cell dies and you lose that stem cell forever. Okay, so why do we need stem cells? Well, is it to contribute to new tissue? Well, there's some evidence to suggest that MSCs can differentiate into chondrocytes and make cartilage. It's still really unclear whether that's happening. But what is more clear is that stem cells and therapeutic cells, regenerative cells, really signal through a paracrine mechanism to endogenous tissue and teach it how to repair itself, give it the necessary tools that it needs, whether it's a new mitochondria or if inflammation needs to be reduced. These cells act in a supporting role, and it's really our own cells that do the majority of repair. So paracrine signaling, that's orchestrated again through exosomes. So you're going to hear perhaps a lot about exosomes as a very hot topic in this area. Again, this is the mechanism by which cells communicate. So if you have a regenerative cell and it goes to the site where repair needs to happen, it tells those cells how to repair by excreting exosomes. And so we can also take those exosomes and use them therapeutically and avoid the use of nucleated cells altogether. Again, that's an FDA concern there. So which exosomes are best for providing pro-regenerative signaling and immune modulation? Well, there are a lot of different exosomes on the market, but our preference at IMAC is for those exosomes that come from a period during intense growth and immune modulation, and that's where the baby is growing, the fetus is growing. The fetus has to modulate the immune system of the mother so that it's not rejected. There's a lot of repair going on. There's a lot of nerve growth, vessel growth, and so products from amniotic fluid are very rich in immune modulatory and pro-regenerative factors from these exosomes, from this heterogeneous population of cells. And so I believe that that's a very strong source. A lot of people ask, well, if you have exosomes, why even use stem cells or therapeutic cells? Well, I like to think of this as a sort of a 1-2 punch, where exosomes are extremely potent at modulating the immune system, but they don't last very long. Once you inject them, they release their goodies very, very quickly. And for the most part, you don't get much transfer of RNA that actually ends up being translated within the host cell. For the most part, the effect is very acute, but it does have a very potent effect. It reduces the immune system, immune response, and reduces inflammation, and gives the therapeutic cells a chance to actually communicate with the host cells. And so what we tend to see with this 1-2 punch is an additive effect, where the exosomes prepare the environment for the therapeutic cells. The cells then are able to communicate with the body, induce regeneration and repair, and over time you then see this improvement of tissue repair. So that's it, and so I'll leave it there, and thank you for your attention.
Thank you, Dr. White. That was a great presentation. I always enjoy when we discuss all the different biologics, the perinatal-derived biologics and their constituencies. And when we look at the biochemistry of cell signaling, so I think that's why exosomes are such a powerful tool to understand, because the biochemistry of the exosomes is in fact the biochemistry of cell signaling. And I think you did a great job at breaking down each of these individual components. I believe there's some 70 manufacturers of these perinatal biologics. There's 1,800 tissue bank certified processing centers. So I don't think that the market is really full of as many bad actors as everybody thinks. And I think the real key to unlocking the power of these products, these tools, comes from exactly what you just taught us. So what they're made of, what they can do, what the basic science behind their behavior is, and some of the translational features, such as the outcomes that you show with your patients, are really leading the way in providing the best standard of care. And I know that you use other technologies to integrate, like health and wellness and diet, exercise, nutrition, and other things to both detoxify, reduce inflammation, and to treat the patient in more of a longitudinal care plan pathway with a personalized approach.
Yeah, that's critical. A lot of people think that cellular therapy is some kind of golden bullet, that you charge $5,000, you give a patient a single dose, and you never have to see them again. That's really not the case. It's been demonstrated that cellular therapy is much more effective if you combine it with IV therapy and other ways to prepare the body for receiving that regenerative medicine. If you inject cellular therapy into a highly inflamed individual with no support from exosomes or any other products, any other preparation, those cells won't survive very long. It's a very harsh environment. So whenever we see a patient that has a major inflammatory component, like the lady that I showed at the end of the slideshow there, we combine exosomes to help modulate the immune response. We give IV therapy that includes a lot of, vitamins, glutathione, B6, B12, these things that really help provide a more conducive environment for the cells to actually begin to function.